Hello viewers, welcome to my channel Biology Notes. Today I will discuss about the structure of G protein coupled receptors or GPCRs, structure of G protein, different types of GPCR, and finally GPCR and G protein activation and their regulation. G protein coupled receptors or GPCRs form the largest family of cell surface receptors. These receptors transduce a wide variety of extracellular signals when they interact with ligands such as neurotransmitters, peptide hormones, growth factors, photons of light, chemical ordants, divalent cations, etc. The interaction between GPCR and ligand initiates a chain of intracellular reactions which ultimately reach the target cell nucleus and alter programs of gene expression. GPCRs are present in all eukaryotes from yeast to mammals. There are more than 800 GPCRs in human have been identified. Now, I want to explain why the name GPCR. If we read the full form of GPCR, then we will understand the fact. GPCR means G protein coupled receptor. It means there are membrane bound receptors which, in active form, coupled with guanine nucleotide binding protein. So, the membrane-bound receptors which are get coupled with G protein in their active state are known as GPCRs or G protein coupled receptors. Now, I will discuss the general structure of the GPCR and the G protein that interacts with that receptor. Here, I have drawn the diagrams to see the general structure of G protein coupled receptors. This is the plasma membrane of the target cell. This is the extracellular side and this is the inner side of the cell or cytosol. All members of the GPCR superfamily share some common structural features. The helices of the receptor span the membrane 7 times. The extracellular domain is for ligand binding. The intracellular domains are for coupling with G protein and interaction with other effector proteins. If we examine more detailed structure, then we will see all GPCRs have the same orientation in the membrane. These are the 7 transmembrane alpha helical regions marked as H1, H2, H3, H4, H5, H6 and H7. These are 4 extracellular loops written as E1, E2, E3 and E4. These are 4 cytosolic segments labeled as C1, C2, C3 and C4. The N terminus is on the extracellular face of the plasma membrane and the C terminus is on the cytosolic face of the plasma membrane. Generally, the carboxyl terminal segment C4 and the C3 loop are involved in interactions with a coupled trimeric G protein. But in some receptors, the C2 loop along with C3 and C4 also involves in interaction with trimeric G protein. Now we will see different subfamilies of GPCR superfamily. According to the differences in amino acid sequence, the members of the GPCR superfamily can be divided into three families: family 1, 2, and 3. Family 1 is the largest group. It can be subdivided into different subsets. The largest subset includes opsins, odorant, monoamine, purinergic, opiate receptors, etc. These receptors have a short N terminus. The second subset includes chemokine, protease activated, and certain peptide hormone receptors. These receptors have a slightly large N terminus. The third subset includes receptors for the large glycoprotein hormones, say TSH, luteinizing hormone, and follicle stimulating hormone. They have approximately 400 residue long extracellular N terminus. Family 2 is characterized by an approximately 100 residue N terminus. Members include receptors for several peptide hormones such as parathyroid hormone, calcitonin, vasoactive intestinal peptide, and corticotropin releasing hormone. Family 3. They have a unique primary sequence, an approximately 200 residue C terminus, and an approximately 600 residue N terminus. The N terminus consists of a putative venous flytrap like domain and a cysteine rich domain. Members include the metabotropic glutamate receptors, an extracellular calcium sensing receptor, and putative taste and pheromone receptors. Here I have drawn the structural diagram of an inactive G protein. G proteins are trimeric and contain three subunits designated as alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid molecules that help bind them to the plasma membrane. In the resting state, when no ligand is bound to the receptor, the alpha subunit has a bound guanosine diphosphate or GDP and is complexed with beta gamma. The alpha subunit binds guanine nucleotides with high affinity and specificity and has guanosine triphosphatase or GTPase activity. The beta and gamma polypeptides are tightly but non covalently associated in a functional dimer subunit. The alpha subunit contains the GTPase domain and binds to one side of the beta subunit. Again, the GTPase domain contains two major subdomains, the RAS domain and AH domain. RAS domain is related to other GTPases and provides one face of the nucleotide binding pocket, and the alpha helical or AH domain clamps the nucleotide in place. The G protein subunits are encoded by three distinct genes. The human genome encodes 21 different alpha subunits, 6 beta subunits, and 12 gamma subunits. Different G protein associate with different receptors. Now, through this diagram, I will show you how the GPCR and G protein get activated and regulated. This is the resting state. There is no any ligand binding to GPCR, hence it is in inactive state. G protein is also in inactive state with GDP. Now, a ligand, say a hormone or an agonist, binds to the GPCR. This binding induces a conformational change in receptor. The conformational changes allow the receptor to bind with alpha subunit of the G protein. This binding releases the bound GDP. Thus, the activated ligand bound receptor functions as a guanine nucleotide exchange factor or GEF. Next, GTP rapidly binds to the empty guanine nucleotide site of the alpha subunit. A conformational change occurs in G protein. As a result, alpha subunit get dissociated from both the receptor and beta gamma dimer. In most cases, G alpha GTP, which remains anchored in the membrane, then interacts with and activates or inhibits an associated effector protein. The beta gamma dimer also sometimes helps in this interaction. In some cases, the activated G protein directly regulates ion channels. Then, the ligand get dissociates from the receptor. The active G alpha GTP state is short-lived because the bound GTP is hydrolyzed to GDP in minutes. This hydrolyzation is catalyzed by the intrinsic GTPH activity of the alpha subunit. The conformation of the G alpha then switches back to the inactive G alpha GDP state, blocking any further interaction with effector proteins. The rate of GTP hydrolysis is further enhanced by binding of the G alpha GTP complex to the effector. So here, effector functions as a GTPH activating protein or GAP. In some cases, a non-effector RGS protein also accelerates GTP hydrolysis. As a result, G alpha GDP quickly reassociates with G beta gamma and the complex becomes ready to interact with an activated receptor and start the process all over again. So this is all about the very general concept on G protein coupled receptors and G protein. If you have any query, then please comment. If you like the video, then please share. Subscribe the channel and press the bell icon to get the notification of new video. Stay with us, keep watching. Thank you.